When Charles Darwin wrote his first book, he concluded that organisms have the ability to evolve. Modern synthesis organized the basic principles that have guided evolutionary biology for the past 50 years. Although biologists knew that forms change, they could not say how that change was accomplished, or how body parts change and new structures arise. Biologists found that evolution is intimately linked with development, the same way a single cell becomes a complex, multi-billion or trillion-celled organism. Basically, all changes in form come about through changes in development. Darwin believed that embryology, a new way to explore biology by analyzing the structure and form of an embryo, was crucial to understanding evolution. He found that genes must be at the center of the mysteries of both development and evolution. The problem was that no one knew which of the thousands of genes in every animal shape its formation and appearance, until geneticists devised schemes to find the relatively small fraction of genes that control the patterning of the fly's body and the formation of its parts. New inventions, such as new techniques for cloning and manipulating genes, and new microscopes, enabled genes to be observed in action. Most of the genes first identified as body-building and organ-forming genes in the fly have exact counterparts in most mammals, and this is helpful in the process of researching structure and form in humans. They concluded that almost all animals share a common toolkit of body-building genes. How could organs of extreme perfection have evolved simply from natural selection? Some thought it was the result of the independent invention of eyes in various animal groups. Walter Gehrig discovered that a gene required for eye formation in fruit flies is the exact counterpart of a gene required for eye formation in humans and mice. The evolution of different eyes has involved a common genetic ingredient. Ernest Mayr thought that very different gene complexes would come up with the same solution no matter how different the pathway was they took. Mayr's theory was proved incorrect. Genomes of different species have been proved to differ vastly. Studies of many animal groups have shown that the diversity arises not so much from the content of the genetic toolkit, but from how it is used. Different animal architectures are the result of applying the same genetic tools in different ways. Vertebrate and arthropod development depends on a set of genes called Hox genes. The genes shape the number and appearance of repeated structures in the main body axes of animals. They determine where various structures form. Hox genes are easily seen expressed in embryos and are responsible for the differences in them between vertebrates and arthropods.
The PAX6 gene in the eye has helped to track the evolution of the eye. Each animal eye has evolved from simpler photoreceptive structures and an ancestor of arthropods, cephalopods, and vertebrae. This ancestor had two kinds of light-sensitive organs and proteins. Shifts in toolkit genes can also explain differences in closely related species or populations in the same species. Evolutionary development shows how patterns evolve. It's easy to imagine that insects invented wing genes, or birds feather genes, or vertebrates hands and finger genes, but there's no evidence that such genes ever arose, but rather teaching old genes new tricks. This happens through sequencing genomes of our own and other species. Darwin's explanation of evolution was founded on the process of natural selection. Evo-Devo shows how complex forms evolve not only in leading one species to the next, but making a body of plans that will affect the higher taxonomic ranks. Macroevolution is the major tenet of the modern evolutionary synthesis that forms above the species level. Microevolution is the evolution within a species. Differences in skeletal patterns of closely related skittleback fish show how the difference in how a single toolkit gene is used. Their different pelvic fin patterns show that they evolved in freshwater lakes. Longer spines show that they lived towards the top of the lake, swimming away from other fish attacks, while the fish with the shorter spines lived at the bottom of the lake, with less threatening predators like dragonflies. In these shorter spined fish, the toolkit genes have been turned off.